Next up, we have Mehdi. Mehdi works in the field of exosomes. He is affiliated, he's a group lead at Sartorius, and he's responsible for upstream processing, downstream processing of exosomes, characterization, as well as analytics. Any one of us who has worked in exosomes understands how difficult all these procedures are, and he's an expert who's going to explain exactly what he does and how he's able to create aids or tools to help us work and do research in exosomes. Over to you.
Um, well, I would like to thank the organizers, first of all, um, for having me and inviting me, especially Bernard. So I thought it might be a good idea to exosomes, or the term extracellular vesicles, a new concept in terms of a therapeutic modality. So I thought it would be a good idea to give a little bit of background on some sort of statistics that you might not have seen yet around exosomes. So I have been in the field of exosomes, or EVs, for around 10 years. I did my PhD around purification, characterization, and therapeutic potentials of EVs. So for this talk, my agenda is going to be a little bit of background about myself. What are extracellular vesicles? What's the definition and the history of EVs? What are some of the guidelines and resources that a new scientist could potentially utilize in their research? As well as what are some of the applications here? And then after that, I'll move to who we are as Sartorius. What do we do in terms of the solutions that we provide? What's the market size as of now for EVs? And what's the expectation in the next decade or so? I'll introduce you to bioprocessing solutions that we have and we work on all the time in a reproducible, scalable manner. And at the end, I'll show you a little bit about analytics and functionality, which are one of the biggest challenges in this field as we speak. About myself, my background is in engineering, so it's a totally different background compared to many of you. So my undergrad degree was in material science engineering, but my PhD was in biomedical engineering from Rochester Institute of Technology. And like I said, I worked on purification, characterization, and therapeutic potentials of EVs, specifically from mesenchymal stem cells. In industry, I've been with Sartorius for around 4 years now. I started as an intern in a team that is called corporate research that I will cover soon. And now I'm kind of involved in all of our efforts around extracellular vesicles with Sartorius. So first of all, what are extracellular vesicles, or EVs? There's a reason that I don't call them exosomes as well, and I'll cover it shortly. So EVs are defined as vesicles that all cell types produce. These are lipid bilayer or membrane-enclosed vesicles that cells produce to technically communicate with each other. They carry RNA, DNA, and they have surface proteins on their surface. These are nanoparticles below around 200, 300 nanometer. So the main point here is that they do not duplicate. So these are what we consider a byproduct of the stem cells, if you will. Where can we find them? Almost everywhere. We drink them in beer, wine. We eat them with cheese. They are everywhere. You can have EVs from any source. You can have them from plants. You can have them from milk. In like human origins, you can have them in all body fluids, including breast milk, saliva, plasma, and serum, as well as conditioned media. So you can easily find them in conditioned media, which makes them a very interesting modality in terms of therapeutic applications. Up to around 20 years ago, we had known these particles with the term exosomes. So we would call all of them exosomes. And it was around 10, 15 years ago that we learned that not all of these particles that cells produce are actually exosomes. So the reason that I don't call them exosomes is because it's almost impossible to distinguish between different subpopulations of extracellular vesicles if you don't capture the moment that they were produced by the cells. And I'll show you shortly. So we came up with a second level of definition which had 3 subpopulations of EVs in it, and they were defined as these 3 groups. So exosomes that are released via exocytosis from multivesicular bodies, and it's a fusion mechanism with the cell membrane. We have microvesicles that are simply budding from the plasma membrane of the cells, and we also have apoptotic bodies that are from the cells that are going, through apoptosis. However, they overlap in size, in density, many other characteristics, physical or biochemical characteristics. So if you purify a sample and at the end of the process look at your particles, it's almost impossible to tell if these are exosomes or these are microvesicles. That's why we call them all extracellular vesicles. However, 2-3 years ago, we actually learned that these are not the only subpopulation of extracellular vesicles. It's even more complicated. And recently, I would highly recommend, if you haven't seen or read this paper and you are interested, this is a great review article learning about lipid droplets, which are not lipid bilayers, they are monolayers. And cells produce them, but also different types of EVs. This includes even smaller particles, as what I just showed you, which we call exomeres, and they can only be purified through a very, very sophisticated process. So the main take-home point here is, if you are going through upstream and downstream processing and purifying them in terms of nanoparticles, it's better to call them, or more reliable to call them, extracellular vesicles. But I want to give you a little bit of history of EVs and how this journey started. For the first 3 decades or 30 years of this field, we thought these were garbage bags. We thought cells were just getting rid of, trash the whole time. And then, so there were studies that they had electron microscopy images of these particles, and then it was around 1981 that somebody defined some of these as exosomes, and they called them exosomes. And then it was around 1983 that 2 studies, 1 week apart, came out, and they introduced the concept of intraluminal vesicles. That means up to that point, we thought all of these were just shedding from the cell membrane. However, then somebody showed these images that they showed no, they are actually getting released from inside the cells, or there is a subpopulation that is being released from inside the cells. Sorry, I'm going to go back because I want to cover one more thing. One other thing is that it was around 1996 that for the first time we learned that these particles do have a biological function. So they are not just garbage bags. It was around 2006 that a very interesting paper came out that they showed that not only they do something, but also they carry genetic materials that can be uptaken by the recipient cell, and they actually do something. They modify the recipient cell. And then all the way up to 2021 that we actually had some clinical trials around COVID treatment using extracellular vesicles. Here are 5 of the examples in different countries that they were initiated at the time for COVID treatment. With the same timeline and guideline, I want to introduce you to a society that was formed that would be very helpful if you are interested to get connected with them and learn about the society. We meet almost every year. So if you look at the timeline that I just described, it was around 2005 that some of the experts in the field, they decided to get together in Montreal. And then 2011, there was another meeting in Paris, and somebody proposed that let's make a society for this. And that's why when International Society of Extracellular Vesicles, or in short ICEV, was formed. And it was around 2014 that the very first paper in terms of a guideline came out to help new researchers, including myself, to learn about what are the minimum requirements to call something extracellular vesicles when you go through a full process. In 2014, they published a paper called Minimal Experimental Requirements for Definition of Extracellular Vesicles. This was very early on. Then in 2018, a second one came out that was minimal information for studies of extracellular vesicles. And it was, I believe, in 2022 that we had an updating myself paper that told us all about the new changes or modifications and guidelines in the field. We also have two different journals coming from the same society. These are very high impact factor journals. One is Journal of Extracellular Vesicles and one Journal of Extracellular Biology. I believe the impact factor of Journal of EVs is around 15 or 16 right now. But I want to show you also some statistics in terms of how many publications have we had in last decade or last 2 decades. So here is a very recent paper in 2020 that covered all of these statistics. So you can see the number of journals and patents is exponentially increasing in the last 5-6 years. And then at the same time, for the first time, we reached almost $400 million NIH funding in 2021 in terms of extracellular vesicle-based studies. If you look at countries and zoom in, United States and China are kind of the leaders in terms of publication. However, at the same time, many of the fundamental and basic research, those very informative studies, many of them, they have been done by European academic groups. However, what are some of these applications? So you can think of, since these are being used for communication between cells, you can think about them as, for diagnostic purposes, for example. As biomarkers. In fact, the only FDA-approved product so far is a diagnostic product in this field. And that is a prostate cancer test kit that you could even, right now, you could buy it and use it for, it's for early diagnostics. And then in terms of therapeutic, you can think about them in two ways. You can think about, could I purify them from mesenchymal stem cells and use their native or natural capabilities, or the other option would be, can I purify them and load them with some sort of drug and then use them for whatever therapeutic application you might have in mind. So these are, again, some of the other statistics around what kind of applications you could apply EVs for. Targeting, biomarker, drug delivery, encapsulation, blood-brain barrier. There are many studies that show that EVs can actually pass BBB. And here you can see that it's also increasing over the last 5 years in terms of number of publications. When we zoom in EVs in therapy, so there are different ways that you could utilize EVs. We know about using cells in therapy. But at the same time, on the other end of the spectrum, we know that we have multiple clinically approved liposome drug delivery structures. So when you look at this spectra, you could utilize native EVs in a natural way. You could engineer your cells to produce modified or engineered EVs and then use them. Or you could not engineer your cells, purify the EVs, and then load them with a specific drug, or decorate the surface of them with a specific protein, if you will. And there is also a new way that is, it's been shown that, so we know how to load liposomes with drugs. There are studies that actually fuse liposomes with EVs because it's easy to load the liposomes with drugs, and then you fuse them. So this way you are using the capability or the potentials of EVs themselves, but at the same time load them with a drug. You might ask, okay, what are the differences between EVs and lipid nanoparticles? This is a fantastic study that you could, it's a review article that you could read that talks about the differences between LNPs and EVs. There are different factors that they consider: complexity, variability, manufacturing expertise, safety, circulation time, and multifunctionality. This is comparing free drug with synthetic nanoparticles as well as extracellular vesicles. Even if you look at the statistics in terms of publications and patents, you can see that, in fact, I mean, it's being called exosomes, but in fact, the number of publications, it has overpassed the number of publications for lipid nanoparticles, and also patents, if you will. How about the stem cell derived EVs, which would be of interest for you guys, I believe. Many studies around the stem cell derived EVs I mean, sometimes it's even hard to find a very reliable one, to be honest with you. But you could use them for different applications. For drug delivery, it could be nanoengineering, drug loading processes. You could, in terms of therapy, you could use them for anti-aging, cardiacs, and anti-inflammation. Wound healing is actually one of the big ones here. It's actually one of the functionality assays. A scratch wound assay is one of the main functionality assays at the end of your process to show that whatever you purified, it's still functional and therapeutic. You can have bioengineering. It's been used for cosmetic purposes as well. So there are a wide range of applications when it comes to stem cell derived EVs. In fact, in the International Society of EVs, there are multiple task forces that you could get in touch with, and one of them is about clinical use of EVs, and to make sure that there are guidelines to make it easier to sit in front of an FDA, if you will. So there are different subgroups even within these task forces. So we want to identify analytical tools for potency assays. This is one of the big challenges when it comes to extracellular vesicles. And also we need to find a way to standardize different EVs to make sure we are getting an enhanced therapeutic potential. In summary, for the first part of my talk around EVs, and this is like, again, all the milestones and accomplishments in the last 50 years, let's say. We thought they were platelet dusts. We started defining EVs and exosomes in 1971, 1961. 1983, we learned that they are actually biological carrier. And then first preclinical study came out in 1998. It was for a cancer vaccine in mice. In 2004, we explored urinary EVs as a biomarker, for biomarker discovery. In 2005, we have two first clinical trials that were reported for EV applications. 2010 was the first time we loaded small drugs inside EVs. Report came out, a preclinical data. And then 2011, one of the main ones that we actually showed a successful encapsulation of RNA inside EVs. And then it was around 2013 that there was a Nobel Prize around protein machinery of how these vesicles fuse with the target or recipient cell membrane and release their cargo inside your recipient cell. And 2014 and 18 are the two main guidelines that I showed earlier that were published for all researchers. All right, now about Sartorius, my company. So we are a biotech company, a global biotech company, around 120 years old. The headquarters is in Germany. We are not biopharmaceutical. We do not enter clinical trials, but we offer technologies, solutions, assays, consumables to all biopharmaceutical companies. We are around now 15,000 people across the whole world. And our headquarters in North America is in New York, Bohemia. And my team and my lab is in Boston. We are located in Boston, which I'll cover shortly. Typically, Sartorius is divided into two divisions. One is called Lab Products and Services, or LPS. The other one is called Bioprocess Solutions. Almost 99.9% of employees are either in LPS or BPS. In terms of what kind of applications they have, so for LPS, these are tools for earlier stage drug discovery and development. And Bioprocess Solutions are more about scalable, large-scale manufacturing of whatever modality you are after. This could be viral vectors, antibodies, proteins, as well as EVs in this case. However, I'm part of a team that is called Corporate Research. So you could think of us as a team one step prior to R&D. So we come up with novel ideas, we do proof of concept, we develop prototypes, And then the goal is to make new modalities more efficient, faster to get to the market. So I was brought to corporate research team with my background in EVs to lead our efforts around extracellular vesicles. A lot of times we collaborate with academia, industry. We do tech transfer at the end. This could be a spin-out company. This could be a licensing in and out. It could be product development. And we also do analyze like new technologies to understand what are the cutting edge technologies for different areas. In terms of market size of extracellular vesicles, in 2020, it was estimated around $170 million. It's expected to have around 28% growth rate and to reach around $2.3 billion in 2030. These are some of the exosome companies, just as an idea. The field is growing exponentially. However, when it comes to bioprocessing and in Sartorius, for different modalities, we offer a complete platform which starts from early stage or upstream all the way to downstream, including upstream analytics, downstream analytics, and the final product, and even functionality assays, etc. So a typical bioprocessing looks like this, and that can be easily adapted for extracellular vesicles as well. We have this, all the bioreactors that you could go all the way to 2,000 liter, if you will, and then all the closed single-use system for buffer, media, etc. Then a typical downstream processing that we apply for many of our purification, it includes a clarification step. Then it follows by what we call tangential flow filtration. Then we do have a chromatography step. The number of steps and the sequence of steps can vary depending what source of EVs you are focusing on. And then we have final filtration, sterile filtration, and the final filling as well. We do offer process development for this. I am not in marketing or in sales team, but I could always connect you with people within the organization who could help you out with these sort of scalable processes. In my team, we focus on analytics a lot. In my opinion, it's the biggest challenge in the field. So there are different ways that you could analyze an EV sample. Overall, when it comes to dose, the most important one would be concentration. What is the number of particles per milliliter that I have in my sample? There has been a lot of different what we call single particle analysis techniques. We use in our lab, we use 3 orthogonal techniques based on 3 different principles to make sure whatever comes out as a report, as a result, it's reliable and it's not an artifact of the limit of detection of that technology per se. One of the technologies is nanoparticle tracking analysis. There are 2, 3 different companies that you could buy these technologies from. This gives you size, concentration of your sample. We do have a flow cytometry-based technique that we use for concentration and protein profiling. If we would like to know what's the percentage of that specific marker, let's say, that is sitting on the surface. And then we also have analytical HPLC, which is a technique that we utilize a lot, and in my opinion, is the most reliable technique in the field as of now. This is semi-quantitative, but it tells you a lot about your nanoparticle sample. This includes what's the protein concentration, not concentration, but like the ratio of protein to particle in your nanoparticle sample. This is just an example. I wanted to put it there because this is how we analyze any nanoparticle sample, including EVs. For example, here, this is conditioned media from mesenchymal stem cells after a clarification step. And what we can do is, even in a crude environment, even in a non-purified environment, we can reliably characterize our EV samples. In the first panel, you can see the size distribution using 2 different principles, scattering and fluorescence of these particles. You can see the number of particles between 2 different techniques and 2 different principles. We have all the proper control experiments that you need in a flow cytometry characterization. And also, we use our analytical HPLC to understand how much protein we have in the background, what are the subpopulation, let's say, because this is coupled with the size exclusion chromatography. Lastly, when you end up purifying your sample, let's say from stem cells, you still, as I mentioned, you still need to prove that these are functional. So one of the very main assays that it's been used for years in the field is simply scratch wound assay. We have a technology that we call InQSite. It's a migration-based technology. And using proper controls, this technology should help you understand if your, let's say, EVs have a better therapeutic impact in terms of how fast your wound recovers. As the very basic standardized assay, you will obviously need more detailed, sophisticated assays later on, animal models, etc., to prove that. But this is automated. It helps you, you can run 96-well plate, 24-well plate, and then you can apply all the proper control experiments, dose dependency, temperature control, drugs that you could use to knock down the uptake mechanism of EVs, and etc. I hire a lot of interns. As a PhD student, I myself started in Sartorius as an intern in the last year of my PhD. So I would like to continue the legacy of interns. So I hire a lot of interns in my team. If you know any PhD students that is interested in this area, I'm more than happy to discuss opportunities with them. These are the last 4 interns that I've had over the last year in my team, and I'm happy to answer any questions.